For most of human history, biological traits were a fixed script. The features dealt at birth dictated an individual's final potential. In 1865, Gregor Mendel proved that this inheritance follows observable rules. By studying pea plants, he identified the factors, what we now call genes, that influence how specific traits pass from one generation to the next. Mendel provided the first map of inheritance, but the code itself remained stagnant. For over a century, humanity lived entirely within the limits of its biological programming. Modern science is moving toward a different approach, treating biological inheritance as a design space. Genetics operates as an information system. Your DNA is a literal code, a sequence of chemical bases, adenine, cytosine, thymine, and guanine, providing the instructions for every living trait. This information-based view provides a framework for three distinct capabilities: reading the sequence, regulating its expression, and rewriting the code itself. Mapping the territory was the first requirement. At the turn of the 21st century, the Human Genome Project reached a massive milestone by identifying 99% of the entire human genetic sequence. This data is stored physically inside the cell nucleus. Information is packed into chromosomes, which act like dense spools of yarn holding the DNA sequence in place. The density of this storage is incredible. If you unraveled the DNA from just one microscopic cell, the strand would be over 6 feet long. Recently, scientists finished the map by decoding the dark regions of the genome, including the centromeres that hold chromosomes together. We finally have access to the complete 20,000 gene instruction manual. This complete map allows scientists to see exactly how the code functions in real time. That real-time control comes from epigenetics. This is a dynamic layer of information that acts as a dimmer switch, turning the expression of specific genes up or down without changing the underlying DNA. The environment communicates with this system constantly. Diet, stress, and exercise act like chemical notes attached to the genes, altering how the cellular machinery reads the script. We can measure the impact of these changes. One study found that walking for an hour a day over 6 months decreased the expression of a major obesity-linked gene by 40%. This mechanism also responds to trauma. Extreme environmental stress, such as starvation, forces the epigenetic system to make negative adjustments to ensure survival. During the Dutch hunger winter of World War II, survivors were limited to 500 calories a day. The epigenetic changes triggered by that famine were passed down, leaving their descendants at a higher risk for depression and metabolic disease. Our habits dictate our current health and set the biological starting line for our descendants. The final phase of this progression is active modification, the ability to rewrite the code. This began with genetic engineering, using bacteria as delivery vehicles. By pasting a human insulin gene into a bacterial plasmid, we turned simple cells into factories that manufacture life-saving medicine for people with diabetes. While effective, those methods were slow. CRISPR changed that. It is the fastest and most precise gene editing tool ever discovered, adapted from a natural immune system found in bacteria. CRISPR uses a guide RNA to find a specific genetic sequence and a Cas9 protein to cut it apart. Once the break is made, scientists can provide a custom blueprint, prompting the cell to repair a defective gene or insert an entirely new trait. We see the results in everything from pest-resistant crops to mice that glow under UV light, providing researchers with a visual way to track cancer and spinal cord injuries during medical research. CRISPR has moved biology from a science of observation into a precise manufacturing industry. These tools offer immense medical potential, including cell therapies that reprogram the immune system to hunt cancer or cure sickle cell anemia. However, these treatments respect a major ethical boundary. Somatic therapies only affect the individual patient. The genetic changes die with them. Germline editing, altering eggs, sperm, or embryos, permanently changes the species, passing those edits to every generation. The pioneers of CRISPR are calling for extreme caution and a global consensus before we cross the line into embryo editing. The code is fully unlocked. As this technology moves into the world at breakneck speed, society must decide what it will write.